firstly for the invitation to speak here, and secondly for so many people still being around. I hadn't in my wildest imagination thought there would be almost a full room at this time in the conference. So what I'm here to do today is basically to evangelize text mining. And the idea is that I don't work on microbiomes, but I know people who work on microbiomes, and they came to me and had a problem. Because basically what's happening for them is along these lines. You know how to do a microbiome study. You design it, you go fetch some samples, you do a lot of sequencing, you have your pipelines, you run it, you get a list of taxa, you have some statistical tests, and you find some things that change significant. So now you have a list of organisms that you found in your samples, and in your case control study, you found that some of them changed. Some went up, some went down, and some didn't change. And that part we sort of know how to do. And then the next thing that happens is that you say, well, now we just need to link this to the current knowledge and write a really nice paper about it. And that's usually where you spend your next roughly 6 months digging around in this. So this is sort of a back of the envelope estimate of the scientific literature. If we think about it very naively, everything is indexed in PubMed. Of course it isn't. And the average paper is 5 pages long. Of course it's longer. But under those assumptions, if you were to print all of them out on standard 80-gram A4 paper and pile it on top of each other, you would now get a pile well over 10 kilometers. In reality, it's probably more than 20 kilometers as well. The thing is, it doesn't really matter whether it's 10 or 20 kilometers. It is painful to try to dig through it. There's just fundamentally too much of it for us to read. So he came to me and said, couldn't you do something with text mining? And I do text mining and other things, so I thought, maybe. But there's one thing, whenever I need to do something like this, which involves getting a computer to do something that even halfway approaches being smart, I rightfully get worried and find it useful to think of the analogy that a computer is about as smart as a dog. By which I mean that if I put sufficient effort into training it, I can teach it to do specific tricks. And borrowing a cartoon from Gary Larson, what we say to dogs, okay, Ginger, I've had it. You stay out of the garbage. Understand, Ginger? Stay out of the garbage or else. And the only thing the dog understood was its own name. And this is sort of roughly my level of ambition when I'm doing text mining. Most of the text is going to be blah, blah, blah to the computer, and every once in a while there's going to be some words that it knows what means. Not the name of the computer itself, but the name of stuff that I'm interested in. And this is what the lovely people doing text mining call named entity recognition, or NER for short. And that may sound very impressive until you realize that it obviously means recognizing stuff with names. And unsurprisingly, when you want to recognize stuff with name, a really good starting point is to have a dictionary of the stuff you want to recognize. So we want to map documents to concepts A and B. To do that, we need to have a dictionary where we know the things we're looking for. We know A, we know B, we know which different names we have for A and B. So what we're interested in this case is, of course, recognizing species, recognizing genera, recognizing NCBI taxa in general. We're interested in recognizing maybe things like diseases, maybe other things like environmental descriptors, ENVO terms, tissues, what have you. Could be functions, could be pathways, could be anything really. And then we need to have synonym information for this. We need to know that, for example, when people talk about this organism and when people talk about that organism, that is in fact one and the same thing. So that's actually where people often run into trouble when they try to do their literature searches. That there are synonyms, you search for a name, you don't find the literature because the literature that said what you were actually interested in used a different name for the same thing. We also want to handle what is called orthographic variation. That's again a text mining term. It's the fancy way of saying that things might be written in slightly different ways. You know, with a hyphen, without a hyphen, variation in capital letters, and also variations like, for example, you have a species name, a Linnaean binomial, and you shorten the genus part to first letter dot, and you want to still recognize it. It's not terribly difficult to do. You just need to have some rules where you teach the computer which variations are allowed, you put it in, you're done. It's just, if you don't do it, you're going to get bad results. The last thing we need is a curated blacklist. And that's a list of the names that we want to block when we're doing text mining. And they're not necessarily in the dictionary because we've done a bad job building our dictionary and we have wrong names in our dictionary. Rather, the problem you have is just that some names also mean other things. And of course, the most notorious problem in that context is three-letter acronyms, or as I like to refer to them, TLAs. And whenever you have three-letter acronyms, you can be pretty sure that the same three-letter acronym can probably also mean something else. And you have to figure out whether the predominant meaning of TLA is in fact a certain organism in NCBI taxonomy, or whether it is a medical operation. So we go through the most common names, the ones that give suspiciously many matches. We look at them and we manually sort out which ones are perfectly fine and which ones are a problem and should go on the blacklist. So once we've done that, good dictionary, a curated stop words list, some rules for doing flexible matching, done deal, we can do named entity recognition pretty well. What can we use this for? Well, one thing is for doing another task in text mining, which is known as information retrieval. And fundamentally, this is the task. You're interested in one or more different things, and you're trying to find documents talking about them. So this might be, for example, you're interested in finding associations in the literature between a certain taxon and a certain disease that you know your microbiome study was about. And the way people normally go about that during the 6 months when they're digging through the literature is what is known as ad hoc retrieval. You know, you go to PubMed, you go to Google Scholar, you type in queries, and the kind of problems you run into is exactly that maybe you're not searching with the right name, etc. Of course, if you have a good named entity recognition method, you can avoid some of these problems with missing papers due to missing synonyms, because you can do NER-based retrieval. And the idea in NER-based retrieval is we run named entity recognition on the entire corpus of text, and then we identify which things are mentioned in which texts, in which documents, and we index that in the database. And then we can search, making use of synonyms, so it doesn't matter whether people use one name or another name for the species, we know it's the same thing. And making use also on the disease side and environment side of ontologies that you have structures, you have related terms. If you're interested in neurodegenerative diseases, you want to find the papers that talk about Alzheimer's disease, even if they do not explicitly say neurodegenerative disease, because Alzheimer's disease is a neurodegenerative disease. So you want to make use of that kind of information, and that would not usually happen if you just do your quick search. So that allows you to find more publications. The next thing is information extraction, linking A to B via documents. We do simple co-mentioning. That works very well. We do some weighted counting to take into account how much do people mention A, how much do they mention B, and how much do they mention them together. And based on that, we get some scored associations that basically give you some sort of statistical property of how much more are A and B mentioned together than you would expect by random chance. And that gives us things like taxon disease associations. It gives us things like taxon environment, taxon tissue, etc. So we can pre-calculate these things. And we can use that for something as basic as doing a term enrichment analysis. Because if you think about the other people doing omic studies, they do a transcriptomic study, they find some genes go up, some genes go down, some genes don't change. You look at the ones that changed versus the ones that don't change, you go to UniProt, you fetch your Go terms, you do an enrichment analysis, and that way you characterize your set. You can of course do exactly the same. Fisher's exact test doesn't care whether you did text mining first. So when I have a set of organisms that went up and a set of organisms that went down, and I have some terms annotated to it, be disease ontology terms or environmental descriptors, I can count, I can do Fisher's exact test, and I can do an enrichment and say, are there some terms that are mentioned together in the literature a lot with several of the species that came out as changing in my study? And that way you can very quickly find what are the overall trends in what I'm seeing. The last thing I want to mention is a little bit of a more outside thing in text mining called knowledge discovery. And that's actually, everything I talked about so far is that you use text mining to find things that are in the literature. And knowledge discovery is all about discovering things that are not in the literature. And you might ask, how on earth can you do that with text mining? If you find something that's not in the literature, didn't you make an error? Well, the idea is you do indirect associations. So the idea is I may be able to link A to B based on literature, I may be able to link C to B based on literature, and based on that I might be able to produce a plausible hypothesis of how A is linked to C via another concept B that I hadn't even thought about. So if you know that you've done a microbiome study of a certain disease, and you know you have certain taxa that came out as significantly changing in that study, you can say, is there something that is associated with both that disease and my taxa that might explain how they are involved in that disease, even if they have never been mentioned in the literature together with that disease. So that allows you to basically discover undiscovered knowledge based on the existing literature. It can allow you to potentially explain observations like I just described. And I think that's potentially a quite interesting technique in this field, even though I have not yet had time to experiment much with it. Lastly, I want to mention that there are some resources out there that you can build on. I'm going to mention, of course, some of my tools, but there are definitely many other groups in the world doing text mining and doing similar things that would be equally applicable to this field. So I've developed a C++ program, Tagger, that does named entity recognition in the literature and does scoring of associations. There's a lot of dictionaries for that, including one based on NCBI taxonomy, one based on disease ontology, one based on ENVO, one based on the tissue ontology, etc. We have pre-computed associations that link these different kinds of concepts to each other based on co-occurrence in the literature. They are weekly updated, so we re-text mine entire PubMed every weekend, so there's a new version going online every Sunday. It's all available under open licenses, so the source code is open, it's available as Docker image. The dictionaries are fully available for download, all the pre-computed results are available. You can find links to all of that on jensenlab.org/resources/text mining. And if you want to see more on what we can do with that in microbiome, we recently ran some workshops on that, and all the training material is online on jensenlab.org/training/text mining. So with that, I just want to acknowledge the people who did the work. So long-time collaborator in Greece, Evangelos Pafilis, who's doing a lot of text mining and working with marine research and so on. So he's a bit more in that direction of text mining, other things than I do. I work at a protein research center. Sune Frankel, former member of my group, who was heavily involved in developing the text mining pipelines we have. Helen Cook did improvements on doing the organism recognition with the Tagger software. Alexander Junge is currently working on developing better scoring schemes for pulling out co-occurrence-based associations. And lastly, Mani, who was the guy who came and asked me, can't you do something with text mining about this? And thanks to the Novo Nordisk Foundation for funding.
We have time for questions. Do we have any questions for Lars?
They are tired after all.
Yeah, it's been a long day, huh? I'm feeling the jet lag, I know that. Oh, wait, here's a question.
How do your wiki updates differ from, let's say, Pubcater has their own things, NCBI photo. How do you think about these two?
So, I mean, those tools are also very good tools, and I know the people behind them very, very well. And in fact, Alexander Junge will be visiting the Pubcater team later this year. So it's a very close-knit community. I think it's very close, the performance of those methods. They also have pre-computed named entity recognition results updated, I don't remember, weekly or monthly or something. It's not really, the important point here is not whether you use their tools or my tools. That's purely a matter of convenience. It's more the overall idea of that you can do concept recognition, you can use very simple statistical methods for linking concepts, and you can use that, for example, for doing enrichment tests. You can use their tools, you can use my tools, you can use other tools out there as well.
I think the other question is, are they different enough to combine them?
No, they're largely doing the same thing. So I would just pick my poison and use one of them. And I think, honestly, I mean, they make different mistakes. I wouldn't say they are better than me or I'm better than them. Text mining makes some mistakes. You pick a tool, it's going to make some mistakes. You pick a different tool, it may have comparable quality and make different mistakes.
I have actually one question, since we ended a little bit early. In order to do text mining, you need a corpus.
Yes, good point.
And unfortunately, because of the way we publish, many of these corpi, is that a word? Corpora, there we go. It's a jet lag, I blame. Are not openly available for people to mine.
Right.
Do you have any comments on how much we're losing as a result of this?
So actually, I didn't talk about this. We published a paper in PLOS Computational Biology a while back looking into this. So comparing doing text mining on 15 million abstracts with the corresponding 15 million full text articles, which were indeed not open access, but via library we got access to them. And it looked like sort of at the same precision cutoff, so the same quality of associations we're pulling out, running exactly the same software, we could get about 50% more. So about 50% better recall at the same precision cutoff by using full text instead of abstracts. So that gives you an idea. We're probably losing about 1/3. That estimate is probably a bit low because you're taking methods that have been tuned mostly on abstracts, and then you just apply them as is to full text without making much effort to fine-tune them to full text. So I'm probably performing relatively better on abstracts compared to full text than I should. So I'm guessing the gap would open up if we do more work on full text mining.
Thank you. That really scares me. I think we need to publish open access.
I think that would be a great idea.
Okay. Thank you.